everyone. This is lesson number 5 of the Synthetic Biology High School Workshop Series. We're covering Foundations of Scientific Project Design. This is part 1 of our last 2 classes, so we're going to be covering more details about this topic in the next section. For today, it's going to be me and Jenny is teaching. Jenny, do you want to introduce yourself?
Yeah, sounds good. Thanks, Miranda. Hi, everyone. I'm Jenny. I'm a senior in Columbia College, majoring in biochemistry, and on the pre-med track, I work in Harris Wang's lab at the Columbia University Medical Center. I'm the founder of this high school workshop series, and I was the former project lead for this group. So for this design, science is very much of an art. Similar to how you train in becoming good at playing a musical instrument or playing a particular sport. So with that, it takes many, many years of training, of practice, of experience to get good at it. And science, it is similarly so. That's why it takes such a long time to become a principal investigator in a lab and to be able to develop your own scientific projects and scientific proposals. So as undergraduates, both myself and Miranda, we will have limited knowledge over how scientific project design works, but we have trained in it quite a bit in our education. And as a result, we're here to present to you the foundations and the very basics of how this design works, so you can get a better grasp of what you'll exactly learn as you learn to become a better scientist. This takes a long time, from your bachelor's to then your PhD, postdoc, and then finally becoming the head of your own lab. So for scientific projects, they are very diverse, but they can be roughly separated into 2 broad categories, and that would be the pure science, as in the theoretical projects, and then there's the applied science and engineering projects. So with these 2 broad categories, there's also interdisciplinary science. And that's when science of many disciplines, this can be many pure science fields or many engineering fields that work together to make the project a reality. And we see this more and more. And we find that generally the most exciting projects is right at the intersection of the different fields of science, whether that be different fields of pure science, applied science, or both. And I also wanted to introduce the idea of interdisciplinary science. So this idea was created more recently in the paper that's shown in that slide. And this idea is that instead of traditional interdisciplinary science, in that, let's say, a geneticist and a computer scientist work together to make a certain type of scientific project that utilizes both genetics knowledge and computer science, you have one person that is able to master both fields, or more than 2 fields, maybe 3 fields at a time, to create their projects. And you see this in biophysics, where biophysics relies on knowledge of both biology as well as physics, as well as mathematics. And this is also becoming more and more common as we see that all of the science skills are intricately linked together. So I would recommend that you read the paper if you have a chance. It's a very interesting idea, and I would agree with the author that this is the future of science, although I think traditional interdisciplinary science will also not go away and is also significant as well. So for going back to the 2 broader categories of science, the pure science and engineering, you approach scientific projects in these 2 fields differently. For pure science, you generally start with a hypothesis. This relies on discovery, understanding how certain principles work in this world. And for both the pure science and engineering, for the purposes of this class, we're going to focus on biology, biological engineering, and biological pure science. So with pure science and within biology, you start with understanding how living systems work, and it's about better understanding how something works. So you start off with a hypothesis, what you think will happen, or something that you've seen happen, but you want to see how exactly does it work. So then you go step by step by step in order to figure out how exactly does the system work. For engineering, on the other hand, you have a pretty good idea of what you want to create in the end. Engineering is about creating something new. So in that sense, instead of having a step-by-step process of investigating properties of a system, as in pure science, in engineering, you have a step-by-step process of accomplishing and creating whatever it is that you want to create. And these steps are ultimately designed differently. So this design process is pretty difficult to talk about abstractly. So I'm going to use two different papers to demonstrate the concepts of generally how a pure science project is designed and an engineering project is designed. So here's the example for pure science, and I will emphasize that for these examples, they are examples, so that as in not all pure science projects are designed exactly the same way, but this example gives us a good idea of the common threads between the designs. We're going to be taking a look at this paper from 1996. So it's a pretty old paper by this point. Induction of apoptotic program in cell-free extract, requirement for DATP and cytochrome C. Don't worry if you don't understand what the title means. We will get through all of it in this lesson. So apoptosis. This is what the authors and the scientific community knew at when the paper was being written. So they knew that apoptosis was some kind of internally encoded suicide program for cells. And in our modern knowledge today, we know that apoptosis is a form of programmed cell death. So this is a way of cells to control which cells die and which cells live. And it may sound pretty awful. Why would cells want to die? But apoptosis is actually extremely important for many different processes. For example, if a cell gets infected by a virus and you don't want that virus to replicate, that cell would want to go through apoptosis and die. Also, when in the human embryo, the human hand originates as a ball of cells. And then programmed cells between our fingers die to allow our fingers to become independent digits. So apoptosis is an extremely important component in our immune system and in development as well as in other areas. And what they knew at that point about apoptosis is that it causes DNA fragmentation. So what this means is that when you look at a cell that's undergoing apoptosis, the long strands of DNA inside the cell start to break apart and they become shreds. And we don't exactly know how that happens yet, so they're trying to investigate how this occurs. They know that activation of CPP32, which is this protein, leads to apoptosis, but they're not really sure what exactly CPP32 does. So the modern name for CPP32 is caspase-3, if you want to look into exactly what this does. They also knew, the scientists at the time also knew that activation of CPP32 occurs through cleavage. So in order for this protein to become active and for apoptosis to occur, CPP32 has to be chopped in half. So their question was, what exactly is needed for CPP32 cleavage and thus apoptosis? So this is a very complicated diagram, and this is an attempt to show you the kind of black box that pure scientists have to peer into to try to figure out what on earth is happening in that black box. So this is modern knowledge of how apoptosis occurs. As you can see, it is an extremely complicated mechanism. At the time, none of these pathways, so that as much as I can remember, either none or very few of these pathways were discovered and mapped out. So this is the kind of system that they're looking into, and they're trying to figure out what's happening. And because there's so many working parts, you really want to concentrate on one protein or a few proteins in particular. And the protein that they will be investigating, cytochrome C, is in the center of this diagram. I don't know if you can see my cursor. Here's an attempt to circle the area of the protein that they will be investigating. So now, what exactly did they do? First, they discovered that apoptosis was initiated by DATP. So this is an ATP molecule, adenosine triphosphate molecule, but the adenosine is D-ribose. So it's like a DNA, it's like a DNA base of adenosine, except with 2 extra phosphates. And they realized that this initiates ADP. And the way they concluded this is that they tested all of these different nucleotides, and all of these are different bases. ATP, CTP, GTP, both the ribose and the deoxyribose versions of them. And as you can see, after they tested all of these here in panel A, only the DATP and the DADP ones give you a cleavage of the CPP32 precursor. So this precursor cleavage is necessary for apoptosis, so they determined ADP was, and ATP, DATP and DADP were involved. And through subsequent tests that I don't show in this presentation, they found out that DATP was the major player. But the point in showing this slide is that they had to go through all of this, test all of these different nucleotides in order to figure out, okay, this is probably the one that's causing the change. And to further substantiate that conclusion, they also looked for DNA fragmentation, another marker that apoptosis has occurred. So in panel B, you're looking at DNA fragmentation in the DATP column and the DADP column, illustrating that they're important for the initiation for apoptosis. And just to give you a better idea what these diagrams are, they're gels. So you can see different bands of different proteins here and different pieces of DNA here. So this laddering effect that you see here indicates DNA fragmentation, and the cleaved products indicated by these 2 arrows indicates the cleavage of the protein. Right. So now that they know that they can begin apoptosis with DATP, now they want to figure out what proteins are involved in apoptosis. So what they did is they did a series of purifications. They separated the cell's proteins using centrifugation and separated them by their densities. And after doing so, separating them into many fractions, they then tested the different fractions that they found. So again, I'm giving a simplified overview of the paper. There are more details that if you would like to learn more, you can feel free to read the paper down here. They found in fractions 2, 3, and 4, there was cleavage of the CPP32 precursor. That means whatever is causing apoptosis has got to be in fractions 2, 3, and 4. Everything else didn't have it. And then if you purify that even further, then you can isolate these specific proteins here. You can see that only 2, 3, and 4 have these proteins, and they're the same size. So they're probably the same protein that came down in different density spins. Now we look at what exactly is this protein. So we have the purified protein in the hands now. What exactly is it? So you can do a spectrophotometric analysis on it. So then this spectrum is shared by the reduced form of cytochrome C. So cytochrome C is a known protein that is involved in OXPHOS, which is a process in cellular respiration in mitochondria. Now this was a little weird. Why would a protein in cellular respiration be involved in programmed cell death? However, the absorption spectrum of that protein indeed resembled the reduced form of the cytochrome C. So now, as you can see, as the paper progresses, they're gathering more and more evidence to try to investigate what exactly is this protein. And they have 100 cytochrome C, but it's rather surprising. So then they tested. Is it that cytochrome C from human cells, bovine cells, which is cow cells, and rat cells have APAF2 activity, which is the activity of cleaving or cutting the CPP32 precursor? And they originally did all of these experiments in human cells and HeLa cells in particular. So as you can see, when there's no DATP, there is no cleavage. With DATP, there is the initiation of apoptosis and thus cleavage. So they thought maybe it's just something weird with human cells. Maybe it's like a weird thing that only human cytochrome C has and maybe nothing other has, and we got the wrong protein. But the cytochrome C from the bovine cells and the rat cells also demonstrate the exact same cleavage upon the addition of DATP. And you know that it must be apoptosis causing this cleavage, is because when you don't have the DATP, there is no cleavage. So now this is gathering more evidence to investigate what exactly this protein is. But it still sounds weird. Like, how is something in cellular respiration involved in programmed cell death? So now they were thinking, maybe our sample just got contaminated with something. When we were purifying that protein, maybe it wasn't actually pure protein. Maybe there's something else in there. So what they did was they tested whether or not the removal of cytochrome C would result in the loss of the apoptotic activity of that cleavage that we were seeing earlier. So what they did is they used monoclonal anti-cytochrome C antibody. And this is a specific molecule that's traditionally produced by the immune system that can specifically bind to certain proteins. So they were using this antibody to specifically remove only cytochrome C from their experimental tubes. So when they found that you depleted the cytochrome C using this method, specific method of depletion, you lost the DATP-dependent activation of CPP32 and the ability to induce DNA fragmentation. So after removing cytochrome C, specifically cytochrome C, you lost the ability to activate CPP32, and you lost the ability to cleave DNA, which is characteristic of apoptosis. So then they were thinking, let's just make sure it was actually the depletion of this cytochrome C that is causing apoptosis not to occur, and not like the antibodies fiddling with something else or getting like stuck somewhere. Once they added back either the purified APAF2, which is the isolated protein from the cells, or they even purchased commercial cytochrome C from bovine heart or rat liver, and they added it back to the extracts, the experimental tubes, it restored the ATP-dependent activation of CPPs, the property of that protein that's causing apoptosis. Then they concluded, in fact, cytochrome C is involved in the apoptosis pathway. So you can see how they started with a hypothesis that they're going to find something in the cells through their purification experiments that would be something involved in apoptosis. And then they took steps to try to figure out what exactly this was. First, through the spectrophotometric analysis to see what it is for cytochrome C, and then to see whether or not this is present in different cells. And then finally, with this antibody depletion assay, to see if you remove it, whether or not the expected effect disappears. So these are all steps that were made and were designed based on the results that you get from a previous part of the experiment. So let's say that when they got their spectrophotometric analysis, this curve didn't actually look like cytochrome C. So in that case, they wouldn't have just done a cytochrome C experiments with a human, bovine, and rat cells to see whether or not cytochrome C was like the factor, because it really depends on what this peak shows you. So in this case, the future steps of a study can be generally anticipated in pure science, but it almost always relies on the previous data that you gather. And that's a marked difference between that method of scientific design versus engineering. So now let's take a look at the engineering design. And again, this is an example, so not all of engineering's experiments are designed this way, but this will yield key insights as to how these projects are generally designed. And in engineering, instead of having future steps directly dependent on the previous steps and what the data shows, there is dependence on what the previous data shows, but there is generally a broad plan that goes step by step on how exactly do I achieve this. Any adjustments that are made are generally troubleshooting efforts to get back onto the original plan. For pure science, because there's so much dependency on data, that plan is often a lot less concrete as to exactly what steps are done. And we'll see this how we have concrete steps in this paper. The synthetic sequence entanglement augments stability and containment of genetic information in cells. So this is a rather recent paper published in 2019. So before we go into this paper, I wanted to review the central dogma, because this is going to be key into understanding how this paper works. DNA is a double-stranded molecule. It is made of nucleotides. So here you can see that there is a single-stranded molecule, and DNA, since you have the U here, this would be the mRNA that's produced after transcription. And then these codons, the groups of 3 bases are made, are grouped together and read together in the process of translation when you convert the RNA into a sequence of amino acids. So in normal translation, you see here, these nucleotides can be grouped in 3 as such. But now you have the exact same RNA sequence here. If you frame shift and shift by 1 base, you can get groupings like so. And notice that this sequence of amino acids is completely different from the sequence of amino acids here. If you move 1 more base over, then you can see that through the translation of the sequence in this frame, you get a 3rd different sequence of amino acids. So as you can see, this single strand of RNA, which comes from a single strand of DNA, the reading of a single strand of DNA, although DNA is double-stranded, can give you 3 different reading frames. These 3 frames are all called reading frames. So if a single strand of DNA, reading a single strand of DNA can give you 3 different reading frames, then the other strand of DNA, because DNA is double-stranded, gives you 3 more reading frames. So for every piece of DNA, there are 6 possible reading frames. And these reading frames are key to what the scientists will do. Okay, so overlapping genes are naturally found in viruses and mammalian cells. And what I mean by overlapping genes is that the start of one gene the start and end of one gene can include the start or end of a second gene, as you can see here. So the plan of this paper is to use cameos, the algorithm constraining adaptive mutations using engineered overlapping sequencings to synthetically overlap genes. That way you can design a specific gene to be overlapped with another gene. And the reason why you can do that is because of the 6 different reading frames that we just talked about. You can have a coding frame in one gene, and then you can have a separate coding frame for the second gene. So you have the 2 genes with 2 different amino acid sequences on the same piece of DNA. Now, why would you want to do that, right? So this project is a biosecurity mechanism. If you can overlay, let's call gene X your target gene, with an essential gene. An essential gene is a gene that if it mutates, if the gene mutates, then the organism will die. So if you overlay these 2 genes together, and let's say you get a mutation in your target gene, and the mutation is a synonymous mutation, so the polypeptide sequence is actually not changed in translation. Because that second gene is in a different reading frame, that will cause a non-synonymous mutation in the essential gene, and the organism will die. So any mutants will automatically be killed. So all the bacteria, and this was done in a bacterial host, all of the bacteria will have less mutations in that target gene. And you can say the same here. Let's say there is a synonymous mutation in essential gene Y. So this essential gene still works fine, but then because gene X is on a different coding frame, this would result in a non-synonymous mutation, so that whatever gene X is probably not going to function correctly anymore. Either way, you prevent any type of mutation to allow this gene, let's say it's a more pathogenic or more dangerous gene, from evolving into a more dangerous mutant. So essentially what this is doing is it's preventing the evolution of your target gene. Now let's look at a second application of this mechanism. So you also have your gene X, your target gene, but now we're encoding it with a toxin gene. So the same thing happens in that if there's a mutation if you overlay these 2 genes, if there's a mutation in gene X that's synonymous, that's going to cause a non-synonymous mutation in. So when you overlay the toxin gene with your target gene, let's say there's horizontal gene transfer. Horizontal gene transfer is the process of having a particular DNA molecule go from one cell to the other cell. And normally, what would happen is that the other cell can take up the genes through horizontal gene transfer, and it becomes part of their genetic material, so they can now express those genes. But if you have that target gene co-encoded, overlapped with that toxin gene, you're going to end up expressing both that target gene and the toxin gene in whatever cell receives the genetic material. So in that sense, any cell that receives the genetic material will be killed by the toxin. But then how do you know that the original cell that has the overlap genes with the toxin and the target gene, how do they not get killed? They don't get killed because there is an existing antitoxin plasmid already in the cell that's constantly producing an antitoxin molecule, and that prevents the toxin from being toxic to the cell. So the cell that you engineer this in is fine. But what this does, it prevents the horizontal gene transfer for one gene to be transferred to the one gene to be transferred from one cell to the other cell. And the process of horizontal gene transfer is especially common in the spread of antibiotic resistance genes. So this is the purpose of creating an algorithm that produces these overlapping genes. And in the algorithm, you input both the gene of interest and the essential gene, and you optimize both the amino acid substitutions, any type of placement, and the long-range interactions in various more complex computational biology methods that we're not going to get into for the sake of this lesson. So here are the steps that I talked about earlier about like the engineering project, as in you have a plan of steps to get you to create what it is that you want to create. So first, you create the algorithm. I'm not going to go into how exactly the algorithm is created, but basically you in the algorithm, you encode for the different types of amino acids and what they're similar to and how they interact with each other. And you try to predict how exactly can I change the amino acid sequence a little bit, but still get the genes to express and function properly. So now that after you create the algorithm, then you start choosing what is the best candidate according to the algorithm for overlapping genes. So you input your two genes, your target gene, and then either your essential gene or your toxin gene, and then you see which type of overlapping, how exactly can you overlap them to best ensure that both your target gene and the essential gene or toxin gene is expressed properly. And you take that best prediction on like the DNA sequence that will probably give you function in both. And then you actually create that in a cell. You take that prediction, you actually put that synthetic DNA into a cell. And in this case, it was a bacterial cell. So now after you put these genes into a cell, then you have to confirm. First, your target gene actually functions properly. Make sure that it's working properly and it is a good reporter. And by reporter, what I mean is a marker that allows you to see the phenotype, the expression of that gene easily. A general good reporter is green fluorescent protein, because if the protein is expressed properly, and it's functioning properly, you'll be able to see that good light. And so you want this good reporter to be generally a good reporter, but also a good reporter for the specific purposes of the project. And I'll go into exactly what I mean about that in a little bit. First, you need to confirm that the first gene functions properly as a good reporter. Then you confirm that the second gene also functions properly, and this gene is either the essential gene or the toxin gene, and it is also a good reporter. So for the specific purposes of the project, you want to test for the stability of the sequence, the ability to prevent mutations, and you also want to test for the prevention of horizontal gene transfer. So what I mean by a good reporter here is that what you choose as your target gene and your second gene, you want to make sure your essential gene is definitely essential, as in mutations definitely will cause the cell to die. But also you want to make sure that that target gene normally is able to mutate. Like for testing 6, stability of sequence, preventing mutations, normally your target genes does mutate at a detectable level that is significant. Because if you find a gene that it just like doesn't really mutate at all, then it's really hard to tell whether or not like your algorithm is working and that you're actually preventing the mutations, or that gene is just naturally immune to mutations. So a good reporter for that first target gene, when you're testing for sequence stability, is that it has to be able to mutate at a fairly significant rate, and you can see a significant dip when you're trying to prevent the mutations with the second gene. So that's what I mean by good reporters in both. It has to be able to allow you to see a significant difference in that when you apply your system versus without applying your system. So as you can see, these steps are general steps that can be created from the very beginning of the project. Before knowing anything about how this is going to end up, you can pretty much already create this plan. compare this now to the cytochrome C paper that we talked about earlier. Without knowing that it was cytochrome C that was in there, we couldn't have known what would be the later test that we did and that we would do for the project testing cytochrome C in human, bovine, rat cells, etc. And my little arrows here is possible optimization. So I say possible optimization because papers don't always list every single optimization step, but you can kind of assume which step that they're optimizing. Sometimes they say it directly in that, like, using these results, then we used the results to optimize the algorithm, et cetera, et cetera. But sometimes you don't. For example, when you're confirming that gene 1 and gene 2 function properly, maybe instead of choosing the one best candidate from the algorithm, you choose a top 5. Because maybe some of the predictions, they don't work, and either gene 1 or gene 2 does not function properly. So then you test all 5 at the same time, and then you pretty much know, okay, this one's going to work. And that's like a step of optimization that is typically not explicitly stated in papers because it's not as essential information as their conclusions. But I just wanted to point that out. There are a lot of optimization steps in typical engineering projects. And just so to wrap up the discussion of this paper, to show you the results of what they come up with, when a sequence of interest was co-encoded with an essential gene in a living bacterium, its evolutionary stability substantially increased. And when a gene of interest was synthetically overlapped with a toxin gene, its horizontal transfer frequency between bacteria was strongly suppressed. So this project was able to successfully create what it had originally intended to create. Now, after going through the 2 major categories of how scientific projects are designed, we're going to go into theory versus reality. And this is about roadblocks in science. So first, we're going to go over roadblocks that you can foresee a little bit more easily, and then there are roadblocks that are a little bit more unpredictable. So in this example, we're going to take a look at the idea of engineering cheaters, a Trojan horse. So the Trojan horse is a popular analogy for a lot of different situations, and it's about pretending somebody is your ally and then like destroying the enemy from within, etc. And you'll see how we're gonna we propose to use this in bacterial communities. So this takes advantage of programmed cell death, that as it which is what we talked about earlier with apoptosis, but this is just considering any form of programmed cell death, in which apoptosis is one form. So when cells undergo stress, cells naturally can go through programmed cell death, in which some of the cells automatically just die. And the reason why they die is because they're able to produce something, some factor, whether it's a small molecule or something else that benefits the remainder of the community. And this is because under these stressful conditions, the bacteria typically don't survive if they all just try to survive. But because a proportion, and it's usually a fairly large proportion of the population, go through programmed cell death, they create this public good, which is then absorbed by the other cells of the bacteria that do not commit this programmed cell death. And then these other cells have a better chance of surviving. So the species of bacteria has a better chance of propagating their own species to make sure that they can reproduce and they won't just all die out. And if you artificially induce stress, you will find that programmed cell death happens at an accelerated rate, and there's an accelerated rate of the production of public goods. So this this idea is that you can use this on pathogenic cells. So imagine how these are pathogenic bacterial cells that cause disease. And if you artificially increase that stress to increase the pathogenic cell death, you get a faster public good generation in the short term and greater fitness benefit for the pathogen in the long term. But what if we engineer cheaters? So cheaters are cells that otherwise are genetically the same as the other cells in the population of that species. They simply are unable to undergo programmed cell death. So when there's programmed cell death in other cells, and then public goods are released, they just take them in, and they just don't die. And these are mutants. So cooperative cells produce public goods, and cheater cells also do not. But they can still exploit the benefits of these public goods. Now, this is a lot of text, so we're going to go through this pretty slowly. If cooperators and cheaters coexist in the same environment, so they equally benefit from these public goods, social evolution theory predicts that the cheaters increase in their frequency and the population become less cooperative. This makes sense. Because if the cheater cells cannot go through programmed cell death, then in their stress, then the cells that die here, all of these cells that die will be the cooperative cells. And by the end, they just keep on dying because of the stress. They keep on releasing these public goods. The cheater cells survive. Eventually, only the cheater cells will be left. And that's why the cheaters increase in their frequency and the population becomes less cooperative. Now, what if these cheaters are less virulent? They don't cause disease as well as the other cooperative cells. So they wouldn't cause sickness as many symptoms. So the frequency of cheaters was negatively correlated with the onset of ventilator-associated pneumonia, suggesting lower virulence for patients with more cheaters. So this is the less virulence of cheaters, but the cheaters still surviving has been already observed. Now, the spatial structure will cause patch-like segregated growth of the cooperator and cheater, so then you have more concentrated goods around and thus more benefit towards the cooperator. So naturally, the species tries not to give cheaters so many public goods, but you can combat this with engineered bacteria in the Trojan horse design. Let's say that these cheaters are cells that you engineered, which they have medically beneficial traits. So there's a couple of medically beneficial traits. We're going to concentrate on reduced virulence and increased antibiotic sensitivity. So reduced virulence, it doesn't cause disease as much. Increased antibiotic sensitivity, it's more sensitive to antibiotics, so it gets killed more easily by antibiotics. Now, you engineer a cheater into the population. You increase the stress. The cooperative cells start dying and releasing these public goods in an attempt to save the species. Eventually, only the cheater cells are left, but the cheater cells are ones that you engineered, so the cheater cells are ones that you know how to kill. In this sense, you can get all of these disease-causing bacteria to kill themselves using programmed cell death, and only have the cheater cells remain, and then you come in with either like with antibiotics or something like that, and you can wipe out all of the cheaters. Now you've killed the entire population of cells that can cause disease. So now let's say it's an antibiotic resistance problem. All of these normally cooperative cells, they're all resistant to antibiotics, but when you engineer these cheater cells, the cheater cells are not resistant to antibiotics, but the cooperative cells don't know that. You increase the stress on the environment, the programmed cell death occurs, the cooperative cells just kill themselves all off because of programmed cell death, releasing the public goods, and only the cheaters remain. But the cheaters are now are not antibiotic resistant, so now you can use antibiotics to wipe out the entire population. So this is one example with the increased antibiotic sensitivity. You can also use that same example with the reduced virulence in the cheaters. So the theory seems pretty perfect. You wipe out the entire population of cells, but there is a fundamental roadblock. Let me go back to this picture. Now, pause the video and try to think about what this roadblock would be. Okay, so that roadblock is the assumption that you can get every single cooperative cell to undergo programmed cell death. In biology, it is extremely difficult to kill off every single cell. That's why fighting cancer is hard. That's why fighting antibiotic resistance is hard. So chances are, even with this method, theoretically it should wipe out everything, but in reality, it probably will not wipe out all of the cooperative cells. Because of that, you have one single cell remaining. That single cell can divide and replicate, and whatever community that you were trying to wipe out before, it can go grow right back. So a possible argument, counter-argument for this is that this wipes out most of the cooperative cells and wipes out most of the virulent cells, and now most of the disease-causing cells are gone, and the rest of the cells, the immune system just takes care of. The immune system couldn't handle it when the population was so large, but now that you've reduced it so much, the immune system can just take care of the scrap and just get rid of all of it, the rest. This counter-argument is plausible, but whether or not this will actually be true is questionable. It really depends on exactly what species of bacteria is infecting the body, and also what the properties and of that bacteria and how bad that infection is, or whatever antibiotic resistant mutant there is. It really depends on a lot of different factors, because there are some species of bacteria in which you only need very few cells. So by very few, I mean around like 100 cells to cause pretty substantial problems. For other species of bacteria, you might need thousands of cells to cause symptoms of a disease. So whether or not the immune system can wipe it out completely and like in time before the bacteria replicate and then regrow that community, that may or may not be possible. So as a result, this is still a fundamental flaw in the problem, in this approach. So this is a way in which you can foresee like that you're probably going to run into issues here. But maybe if it's something where you're just trying to reduce a population, maybe this theory would work very well. Now, again, let's look at theory versus reality. Where are roadblocks more unpredictable? In this example, we're going to take a look at DNA single base editing. So most genetic diseases arise from point mutations. Point mutation is a mutation in a single nucleotide of DNA. And current approaches to correct point mutations are inefficient, and they typically induce many random insertions and deletions, also called indels, at your target DNA sequence. These insertions and deletions are typically caused by the fact that there is a double-stranded break made with the enzyme in trying to make that correction, and then the DNA repair system starts to get involved, and it realizes the DNA is damaged, and then you start having these insertion deletions because the repair system doesn't totally know like what is going on. It's trying to fix it too. So the base editing enables the direct and irreversible conversion of one target DNA base into another in a programmable manner without requiring a double-stranded DNA backbone cleavage. So the cutting of DNA of the backbone of the DNA, or a donor template, a template DNA that is used to try to make a particular genetic change. So it's generally much easier to create very large DNA edits compared to small DNA edits, and this is the small DNA base editing is what we're going to be looking at today. So the DNA editing mechanism relies on CRISPR-Cas9. So I just wanted to give a quick introduction to this. CRISPR stands for clustered regularly interspaced short palindromic repeats. They were how these systems were discovered in the bacterial immune system, in which you have these like repeats that contain certain instructions for proteins to cut specific piece of DNA. So the Cas9 is a protein, an enzyme that cuts DNA at a target that is encoded by a guide RNA. And this guide RNA is here. So the guide RNA loops into the Cas9 protein, but also matches with the specific DNA target. So it can cut here. So just for fun, there is also a nuclease dead Cas9 that can bind to the target DNA, the target encoded by the guide RNA, but does not cut. And the for fun was a joke. In fact, this is how exactly you direct a specific type, like a protein to a particular region. So you can program this to go wherever the guide RNA wants it to go, and then you can fuse this protein with other proteins. So basically you can direct where other proteins go by fusing that other protein with the Cas9. So we're going to look at two different base editors here. So the first one was the base editor that was a little bit easier, comparatively speaking, to create. And that substitution is going from a GC to an AT. So the way it was done is that you fuse that nuclease dead Cas9, which directs the Cas9 to a particular location because of this guide RNA. You fuse that Cas9 with the cysteine deaminase. So basically, wherever this dCas9 is programmed to go, the cysteine deaminase is now going right with it because it's connected. Now, what does the cysteine deaminase do? So the system, the deamination of cytosine is catalyzed by cysteine deaminases and results in uracil, which has the base pairing properties of thymine. So it basically changes a cytosine into a uracil, which acts like a thymine. Now, so the cytosine deaminase is coming in here, and here's your target. This is the GC pair that you want to do the edit of. Now, it takes out, the cysteine deaminase takes out the C and replaces it with the U, which, it takes out the C here and then opens it here. And then it deaminates the target C and turns it into a U. And then when you get DNA replication or repair, it gets repaired as a T paired with an A. So then you get a GC paired with an AT. And you may say, but this is only going to change C, it's not going to change G. Well, if you want to change G, then you just change the C on the other strand of the DNA, and then you can change the G. So then after fusing the dCas9 with the cysteine deaminase, the group then did some protein engineering on the cysteine deaminase to then optimize this activity of switching out the C for a U, and that gets converted into a T. For the second base editor, now you've done GC to AT, now you want to do AT to GC. How do you do it the other way? And the importance of this is just shown in this figure here. in which you can see that most of the pathogenic for disease-causing human SNPs, or single nucleotide polymorphisms, which is the error or the difference in a single nucleotide, most of it, 48%, would be corrected by the AT to a GC mechanism. And this is exactly what this base editor seeks to do. So in this sense, you have an A. With the A can change into a different form. This is the tautomerization. With the removal of the ammonia group, and you can turn it into an inosine. Now this inosine base is read as a G by the polymerases, so DNA or RNA polymerase. So what they want to do is, again, you can see that this engineering project, here's the plan. You have a Cas9 nickase, and then you open that DNA, and then you have some kind of hypothetical deoxyadenosine deaminase. And the deoxyadenosine deaminase simply means an enzyme that does this. So then with the binding and the genomic DNA, opens the DNA, and now you see that A and T is revealed. That A is then turned into an I by this hypothetical enzyme that hasn't been created yet. And it deaminates that A and turns it into the I in that bubble here. And then through DNA repair and replication, it's read as a G, and here is a C. So then you've turned this AT pair into a GC pair. Now, how do you create this hypothetical enzyme? So what you really want is an adenosine deaminase that processes DNA. But there are no enzymes that are known to deaminate an adenine in DNA. So notice how this differs from the cytosine deaminase here. Most cytosine deaminases actually edit RNA, but there is a cytosine deaminase that already edits DNA. So they were able to directly fuse it and then do optimization on it. But the situation is different here because there are no enzymes that actually do what you want that hypothetical enzyme to do, which is edit the DNA, converting AT to GC. All of these adenine deaminases, which are enzymes that turn that AT into a GC, work on RNA rather than DNA, and they're not effective on DNA. So what the lab did is that they evolved this adenine deaminase that accepts DNA as a substrate. So then you can get the AT to GC substitution on DNA. Now, to enhance this system, for base editor 1, so base editor 1 is the one from GC to AT with the fusion of the cytosine deaminase. In addition to fusing that cytosine deaminase, they also added BE2B3, which is a protein that increases editing efficiency. And the reason is because, now, when you have cytosine deamination, you turn that C into a U. But normally, the cellular uracil glycosylase, the uracil DNA glycosylase, it catalyzes the removal of the U in DNA because uracil is not supposed to be in DNA. So the cell knows it's a mistake. And it initiates base excision repair. So all that work you put in to try to get a U, to get that base change, it's gone because this glycosylase over here snips out that U and changes it back to CG. Now, we don't want that to happen. So we found, the lab found inhibitors, BE2, BE3, to block the activity of this glycosylase. So now that you're blocking the activity of this, you allow for this to proceed where you have a UG and then it turns into a UA and a TA. And here, the nicking by BE3 and the cellular mismatch repair, we're going to come back to this. We're going to first talk about it in the next slide, this slide. So you also add a nickase for editing efficiency. And the nickase is added for both the base editor 1 and the base editors 2, and we'll see why. So the eukaryotic mismatch repair uses these nicks And nicks are a cut in a single strand of the backbone of a single strand of DNA. So not a double strand, just one strand of DNA. Eukaryotic mismatch repair uses nicks present in the newly synthesized DNA to direct the removal and resynthesis of the newly synthesized strand. So what this means is when you have this mismatch repair, you have a U and you have a G. Clearly that's a problem because this U also looks like a T. So it's going to pair with an A, and the cell thinks like, oh, I screwed up here somehow. So I want to change it back to what it used to be. But then how does it know which is the new base? Which was the error base? Was it the U or the G? And if you think back to DNA replication, so the leading strand usually occurs in several different fragments where you have the helicase unwinds the DNA, and then you have the synthesis of the leading strand, and then with the lagging strand, you have Okazaki fragments, and then helicase unwinds some more. And basically, because of the Okazaki fragments, there are existing nicks before the ligase comes together and then seals all of different fragments of DNA together. As a result, the cell knows the newly synthesized DNA is the sequence of DNA that has more nicks on it. Now, you spent all that time trying to turn the CG into the UG. Now you have the UG, but then you have a nick because then you have the cytosine deamination. But then the cell is going to try to see like, oh, this U is wrong. The G is the old strand, the U is the new strand. Let me turn it back to a C because that's the old strand. You don't want it to do that. So then you can add a nickase. You also fuse a nickase to the Cas9 so that the nickase creates DNA nicks on the other strand of DNA, the strand that you're not editing. So then when the cell looks at this again, they're saying, ooh, this strand has more nicks. This must be the new strand and this must be the old strand. The G must be part of the new strand and the U must be part of the old strand because the strand that has the G has more nicks. And therefore, what I'm going to repair is the G and not the U, which is exactly what you want. So then the B3 favors repair of the nick strand. So it changes that G into an A. And then the desired outcome comes out with a TA. The other possibility, CG, is not favored because the G is the one that has more nicks. And there you have it. You have a conversion of a CG to a TA. So the reason why the previous one, the B2B3 and the blockage of the uracil DNA glycosylase is only for base editor 1 is because this specifically works on the removal of a U, and the U is not involved in the second base editor from AT to GC. It's only involved in the first. But the nickase, however, whenever you do this, you always create a small nick in the backbone, the DNA backbone. As a result, you have this nicking pattern in both the GC to AT base editor as well as the AT to GC base editor. So that's why you want this nickase to be part of both. Nicking the DNA strand containing the unedited G would simulate newly synthesized DNA so that G is edited. So this design is honestly really brilliant, and all of the plans, all of the optimization that went into it is extremely, extremely brilliant. But even this very good design, where you can't whenever you have enzymes doing something, whether it's this nicking or it's restriction enzymes, cutting a specific site, you always have something called off-target effects, or in the case of restriction enzymes, it's called star activity. It's basically stuff that's not supposed to happen. So in this case, off-target effects would be, go back here, you're trying to direct the Cas9 to a specific target. Sometimes the Cas9 goes to a different place in the DNA and doesn't actually go to a place that's your target. And any activity it has in different areas of DNA is then called off-target activity. Similarly, with restriction enzymes, with star activity, sometimes you get the restriction enzymes don't actually cut at a restriction site; they cut at a different site. And for restriction enzymes, it's actually the enzymes have been enzymes that's used in the lab have been optimized enough so that the star activity is quite rare. In this case, with this new system of the cysteine deaminase, the different base editors, it's hard to say exactly how significant the off-target activity is. So this is a case where you're not going to know how much off-target activity you're going to get until you actually create the system. But this is a risk, and then we'll take a look now at these unintended consequences, this off-target effects.